So today we're talking about treatment of infection. So just rounding up the subject by looking at how do we actually treat all these nasty infections that we've been talking about earlier. So the first question is, how do we actually achieve treatment? How do antimicrobial agents actually work? And the key concept at the heart of all this is selective toxicity. So what we look for are agents which have an effect on the microorganisms we're targeting, but don't have an effect on our own cells. So there are plenty of substances out there which kill cells, all kinds of cells, but we don't want that. We want those which will target, either block irreversibly or just inhibit some kind of pathway or other piece of biochemistry, which is only in the microorganism and not found in us. Or sometimes we do target things that are conserved in both settings, but they're actually sufficiently different in the microbial setting that we can use them as a target, for example, the ribosome. So one term that's sometimes used for what we're looking for here is a magic bullet, which kills the bacteria but doesn't kill the host, or kills the viruses but doesn't kill the host. Now, another important concept we have to get across is that different agents have different spectra of activity. So not all antibiotics work the same, not all antibiotics target the same bacteria. They target different kinds of bacteria. So sometimes we use the term narrow spectrum to describe an agent which only targets a very small number of organisms. So the very first penicillins were like this. They had good effect against, for example, Streptococcus pyogenes, but were fairly useless against things like E. coli or Pseudomonas aeruginosa. Since then, we've actually broadened the spectrum of these kind of antibiotics, and we now have the so-called third generation cephalosporins have very, very broad spectrum. We'll target gram-positives, gram-negatives, they'll target E. coli, Staph aureus, Pseudomonas, and so forth. Because of the difference in the way the cell walls are made, some antibiotics will only work against gram-positives, and these are particularly large, bulky molecules like vancomycin, which can't get across the gram-negative outer membrane, but they do very effectively interfere with cell wall biosynthesis in gram-positives. There are also some that only target gram-negatives, in fact one that I know of, aztreonam in particular. Now it's worth stressing that antibiotics don't work against viruses and fungi. So you've probably heard about this many times before, when the GP says, oh, there's no point in giving you antibiotics for a cold, because the antibiotics target bacteria and the cold is caused by viruses. Now that's not to say that there aren't specific antiviral agents. There are none that are active against the cold virus, but there are antiviral agents. There are specific antifungal agents and antiprotozoal agents as well. But generally, the agents we use are targeted to one particular group. They don't work across all of them. Now, when we're looking at antimicrobial agents, particularly antibacterial agents, we can classify them as bacteriostatic or bactericidal. This is an important distinction. So bacteriostatic agents just inhibit the growth of the bacteria. So the bacterial cells just stay there, roughly equal numbers, just hunker down, they don't grow, but they don't die either. Whereas with bactericidal agents, we're actually actively killing those bacteria. Now, we've already mentioned the diffusion test in a previous lecture, where you have these zones of inhibition around discs that are put on the plate, and you can measure those, and that will give you a measure of the susceptibility of the organism to the agent. And then we have the MIC as measuring this much more accurately, giving us a concentration of antibiotic which is the minimum necessary to inhibit growth. Now you can also use this similar test to look at the MBC, or minimum bactericidal concentration. So if you took all those tubes there, the cloudy ones obviously got growth in them, but the one that's not, the couple next to that in terms of the doubling dilutions that don't have obvious growth, if you plate those out, if this was a bacteria static agent, you may well see some growth in those tubes. So it hasn't killed the bacteria. And the MBC is which of those tubes, the first of those tubes where you actually kill the bacteria in that tube and you don't get any growth. Now, in terms of the history of synthetic antimicrobial agents, these were the first agents we had, again, active against bacteria. And the German microbiologist Paul Ehrlich initially began studying this idea of selective toxicity in the early 1900s. And he was looking at the idea that if you can stain the bacteria with dyes, then maybe you can use those dyes to inhibit their growth. And he came up with this drug salvarsan, one of the first effective antimicrobial drugs. And these agents generally are growth factor analogues. They're structurally similar to the things that the bacterium needs to grow. They get incorporated into the cell. They get into the cell. They get into enzyme active sites, but they don't function properly, and they inhibit those pathways. And there are many examples of that. Sulfur drugs were invented or discovered by Gerhard Domack in the 1930s, and these inhibit the growth of bacteria static agents. Sulfanilamide was the simplest and the earliest one. We also saw a few years later, in the 1940s, isoniazid coming into clinical practice, which is a growth factor analog effective only against mycobacteria, against particularly Mycobacterium tuberculosis, the agent that causes TB, and it interferes with the cell wall there. And then more recently, in the late 70s, early 80s, but probably in the early 80s, I guess, the quinolones came in, antibacterial compounds that interfere with DNA gyrase. And again, they are wholly synthetic. Now, if you are going to be strict about the use of terms, we have to make a distinction between antimicrobial agents, antibacterial agents on the one hand, and antibiotics. So the term antibiotic is applied to naturally occurring antimicrobial agents produced by bacteria or fungi out there in the natural world. And they're part of the ongoing war between different organisms out there to occupy different niches, different environments, the struggle for existence that goes on in the soil and in waters and so forth. And there are many, many antibiotics out there that you can isolate. Very few of them are actually useful clinically because many of them are toxic to humans as well as to bacteria. But there are a small fraction of them that are actually useful. And the useful ones, what we've done with those is we've enhanced their efficacy by taking the original chemical structure and modifying it using a synthetic chemistry approach. And so we sometimes call those antibiotics semi-synthetic antibiotics. So the original starting material comes from nature, but we modify it. Now, I borrowed this from Wikipedia to try and keep these figures in the public domain, stuff that's in the public domain. It's a very busy slide anyway, but it just gives you a feel for the range of targets of different antibiotics. So many of them are targeting cell wall biosynthesis, some of them attacking the cellular membranes. The other major group are those that target protein synthesis and target the ribosome. And we'll say more about some of these in a few moments. So if we look at antimicrobial agents that act on the bacterial cell wall, most of these actually interfere with the synthesis of this peptidoglycan layer. So most bacterial cell walls have this kind of molecular chainmail, where you basically got this one molecule that is huge in its extent, encompassing the whole cell, with these long polymers of NAGNAM, N-acetyl uramic acid and N-acetyl glucosamine, cross-linked by these peptide bridges. And antimicrobial agents act here, eventually cause cell lysis. They eventually cause this chain mail to break open, and because of the osmotic pressure in the cell will actually burst out and die. They tend to bind to specific enzymes actually responsible for the creation and maintenance of this peptidoglycan layer, otherwise known as penicillin binding proteins, or PBPs. Now, the most widely used group of agents that act on the cell wall biosynthesis, are the beta-lactam antibiotics, and they're probably the most important group of antibiotics. They include penicillins, cephalosporins, and the kefamycins. And in broad terms, if we look at the structure, chemical structure of these, we can say they look a bit like a house with a garage stuck on the side. And the garage there, square bit here, is the beta-lactam ring, which gives them their name. And you can see that there's a satellite dish stuck on there as well. That is the kind of place where we will add modifications, chemical modifications, to the basic beta-lactam ring to give us antibiotics that have different properties, that maybe can be taken orally, or maybe they have a broader spectrum than what we started with. Now, the most important group of beta-lactams are the penicillins, discovered by this guy here, Alexander Fleming. Although that's a short answer. So actually worth looking. The Wikipedia is a mixed blessing. There are lots of things on the Wikipedia you don't trust. But interestingly, there's a whole page on the Wikipedia about the discovery of penicillin before Fleming, and that lots of other people, they were kind of prequels to the story where other people got there first. But it was Alexander Fleming that actually discovered it and then took it forward, and it eventually moved into clinical practice. So this is primarily effective against gram-positive bacteria, as I mentioned, things like Streptococcus pyogenes, Streptococcus pneumoniae. In the early days, even Staphylococcus aureus was sensitive to penicillin. But it took these chemical modifications to actually make it more broadly, this class more broadly usable, and we now have synthetic forms that are active against a wide range of gram-negative bacteria, from E. coli through to things like Pseudomonas aeruginosa, all active, all now susceptible to beta-lactams. The second group of agents within the beta-lactam class are the cephalosporins, and these can be divided into those that are orally active and those which have a broad spectrum, but which tend to have to be given intravenously, or at worst intramuscularly. These are like a house with a garage and a basement. So we've got a basement on the bottom of that ring there. And here again you've got these two different positions where you can add all sorts of side chains to change the properties of the agents. And some of these, like ceftazidime, now have very, very broad spectrums indeed. We also, there are some unusual beta-lactams that have been derived by the pharmaceutical industry, so-called carbapenems, imipenem and meropenem. These look like a house that's kind of undergone a bit of subsidence. Maybe it's been built over a mine shaft or whatever. These have a very wide spectrum of action. These are the kind of equivalent of domestos for the worried doctor on the ward. So when a patient's going downhill with infection and they don't know what to do, particularly a critically ill patient, they will give them one of these carbapenems. Typically nowadays meropenem is used in that circumstance. Monobactams is this where they just consist of the garage, if you like, nothing else. Aztreonam is the only example of that that's seen widespread use, and even that's not particularly well used, but that's used for gram-negatives only. So that was antimicrobials acting on cell walls. As I mentioned, the other major class in terms of size and importance are those that act on protein synthesis, and particularly on the ribosome. Now, the bacterial ribosomes divide into these two subunits, the 30S and the 50S subunits. They differ slightly in size from the ribosomes that are found in your cells, and that's why they provide a target for selective toxicity. We have a whole range of agents here. I'm not going to rehearse them all in detail. The minor glycosides, these are given intravenously, but include things like streptomycin, gentamicin, amikacin, and the tetracyclines, both binding to the 30S subunit. 50S subunit, we've got chloramphenicol, fusidic acid, and then a class of agents known as the macrolides. Some of you may have taken erythromycin when you've had sore throats and things like that in the past from that group. There are some new agents that have come on the market in the last 10-15 years. Daptomycin is one, produced by Streptomyces and acts on gram-positives. Platensimycin. This is a new class of agents which actually blocks a new target, fatty acid biosynthesis. And this appears to have a very broad spectrum. So there is some progress in this area, although it has to be said that there are fewer of these agents than we'd actually like. There are some antimicrobials that act on nucleic acids. We mentioned them briefly. Quinolones we've already mentioned. Metronidazole, which is a broad spectrum antibiotic in one sense, in that it targets all anaerobes pretty much, but it's narrow spectrum in another sense because it only targets anaerobic organisms. But so metronidazole is very commonly used in this country if we suspect there's anaerobic involvement. So if someone has an abscess in their liver, if they have leakage from the bowel where there's likely to be anaerobes getting into the abdominal cavity, then this is where we would reach for metronidazole. And there are some agents that inhibit transcription. Rifampicin, which is a key drug used against tuberculosis, inhibits a bacterial RNA polymerase. And there's an agent, flucytosine, used against yeast, which also acts as a kind of analogue of a base incorporated into ribosomal RNA. So when we talk about antimicrobial agents, unfortunately we have to very quickly move on to a big problem with the use of antimicrobial agents, and that is the problem of antimicrobial drug resistance. So this is the ability of microorganisms to shrug off the effects of antibiotics and other antimicrobial agents. In some ways it's not surprising, because for antibiotics, those antibiotics have been around for millions of years, billions of years. They're part of the struggle for existence in the soil. And the organisms that make the antibiotics have to have their own ways of avoiding being targeted by them. And so many of the resistance genes we see causing problems in the clinical arena have actually come from that kind of setting. And there was a paper in, I think it was in Nature last year or so, where they actually got some ice cores that are 30,000 years old and managed to find antibiotic resistance genes in them. And you sort of think, well, that's actually not that surprising, but they managed to get a high impact paper out of that. What is the problem? Well, the problem is that the use of antibiotics in medicine, in veterinary practice, in agriculture, is bringing forward Darwinian selection of antibiotic resistance. So the more we use these agents, the more we're actually applying selective pressure on the microorganisms out there to become resistant. There's a lot of controversy here about whether uses of antibiotics for things other than human infection are justified. There's also a lot of controversy about whether most antibiotic usage is actually justified even in treating humans. So people do go to their general practitioners with a cold, feeling miserable, and sometimes they do pressure the GP into giving them antibiotics, and if they're a troublesome patient, the GP may sometimes give them antibiotics. And so this is why you see these kind of campaigns here, to try and persuade people you don't need antibiotics for colds and flu. You should just go home and take some paracetamol. There are questions about, we can actually, if we use antibiotics properly, then we can minimize the incidence of resistance. Sometimes the word stewardship, antibiotic stewardship, is used to describe the appropriate use of antibiotics in hospitals, and actually not using antibiotics when you don't need to, using them for the shortest time possible, using narrow spectrum agents rather than broad spectrum agents. All of these things can be done to try and prevent resistance from being selected for. Problem is that it's not straightforward resistance anymore. We actually have multi-drug resistance as a very common phenomenon. So many bacteria are actually stacking up multiple resistance determinants and becoming resistant against all sorts of things. Some of those organisms that we see that are multi-drug resistant in the hospital are just inherently multi-drug resistant. They've always been that way. Xanthomonas multifilio is one, for example. They're also, though, common or garden bacteria, things like E. coli and Staphylococcus aureus, that have steadily acquired resistances over the last few years, over the last few decades. And you may have seen in the newspapers a couple of weeks ago, one of my colleagues, Peter Hawkey, stressing the problem of multi-drug resistant E. coli in our communities here in Birmingham, and the spread of that agent, of that organism, from the Indian subcontinent into the heart of Birmingham. So how do organisms become resistant to antibiotics? There are various mechanisms that they can deploy to become resistant. So they can do away with the structure that the antibiotic inhibits. So if it's not there, then they are just inherently resistant. So something like isoniazid is not going to have any effect on many organisms because they just lack the target. The organisms can be impermeable to the antibiotic, so the outer membrane of gram-negative bacteria provides quite a barrier. Sometimes the thick peptidoglycan in gram-positive similarly provides a barrier. Many organisms produce enzymes that inactivate the antibiotic. So we have beta-lactam antibiotics, and we have beta-lactamases, enzymes produced by bacteria which break down those beta-lactam antibiotics. The organism may modify the target of the antibiotic or develop a resistant biochemical pathway. So MRSA, for example, what that organism has done is it's acquired a gene for an additional penicillin binding protein which is resistant to antibiotics like methicillin. It allows it to still build its cell wall with this so-called PBB2 primed, but that enzyme is now impervious to the effects of the antibiotic. And the other thing that happens is that many bacteria develop pumps, so they can just efflux mechanisms, so they can actually just pump out antibiotics out of the cell. So although if the antibiotic accumulated in the cell to a sufficient level, it may start inhibiting targets in the cell, it never gets there because it's just been pumped out as quickly as it's getting in. Now, how do we get resistance in the first place in terms of genetics? Well, there are two ways in which this happens. Now, one way is you can get the steady accumulation of mutations in the chromosome of the bacterium. And you can get selection for resistance within a particular lineage, within a particular chromosomal lineage, happening in a sort of stepwise fashion. That's very common in tuberculosis. So in Mycobacterium tuberculosis, if you had tuberculosis and I gave you a single drug, let's say I gave you streptomycin against your tuberculosis, about half the time you would relapse with a drug-resistant strain. If we sequenced its genome, we'd find that it would have 1 or 2 point mutations in the genome which were enough to give it resistance. Similarly, in viruses, if you were infected with HIV and we gave you a single antiretroviral agent, there is a very good chance that you would relapse over time with a drug-resistant strain which had accumulated a few point mutations in the active site of the enzyme that you're targeting. Unfortunately, in addition to that approach, bacteria have harnessed another approach, which is that they use mobile genetic elements. So instead of these mutations occurring in a lineage, you actually get the transfer from one lineage to another en masse of whole genes, sometimes whole gene clusters, moving through what we call horizontal gene transfer. And one of the terrible effects of this is that you don't just get the transfer of one resistance determinant at a time. They're often stacked up. The genes are actually clustered together. So, for example, here's a plasmid, a small piece of circular DNA which can replicate independently the chromosome and can move from one cell to another independently. And on this plasmid here, you can see we've got chloramphenicol resistance, kanamycin resistance, sulfonamide resistance, ampicillin resistance, tetracycline resistance. And so we have a problem here that if we treat with any one of these agents, we're going to select for a population that is resistant to all of them. And many of these resistance determinants, these mobile genetic elements, are able to move across quite considerable taxonomic distances. So they can move from gram-positives into gram-negatives, or vice versa. So this is where we have a big problem. We're actually this kind of ratchet effect. As we use antibiotics extensively, we select for these kind of elements in bacterial populations, and we end up with multi-drug resistance. Even when we're using a single agent, we can select for a multi-drug resistant organism to come out of that. So why should we care about antibiotic resistance? Well, there are various reasons why this is a problem to us. Things that used to be simple to treat are now much harder to treat. So back in the 1950s, Staphylococcus aureus, if you had some abscess, wound infection, carbuncle caused by Staph aureus, it would respond to penicillin, very straightforward. Similarly, if you had pneumonia caused by Streptococcus pneumoniae a few years ago, you could reliably have given penicillin and it would have cured it. Now we have to give agents which are much more expensive, typically have to be given by injection rather than orally, to be given for longer, and it just makes the whole thing harder. For MRSA, we have to give vancomycin, which is a nasty agent which you have to give intravenously, and it has certain, if you run it in too quickly, it can produce toxicities, this syndrome called red man syndrome. We are actually, in certain circumstances, approaching what we might call the post-antibiotic apocalypse, where we actually have no antibiotics, no antimicrobial agents left capable of treating a particular bacterium. It's an organism we study in my research group called multidrug resistant Acinetobacter baumannii. There are cases where this organism is resistant to everything. Similarly, you may have heard recently with tuberculosis, we've had multi-drug resistant tuberculosis for a long time. We've had extremely drug-resistant tuberculosis for a few years, and now we're seeing completely resistant strains, pan-resistant strains of M. tuberculosis. Luckily, so far, we tend to see these problems either in restricted areas, not spreading across the globe, or in low virulence pathogens. Acinetobacter baumannii is not damaging to anyone here. It's an opportunistic pathogen. It will only target very ill patients anyway. But there is this concern that maybe one day we will start seeing multi-drug resistant organisms coming to the fore. We're already seeing it a little bit with MRSA in the community. We've finished with bacteria. Let's now just quickly talk about some other agents. Antiviral agents. So antiviral drugs, problem is that they have to, viruses in effect have a eukaryotic lifestyle. They parasitize our cells. They're using a lot of the machinery inside our own cells as part of their life cycle. So it's quite hard to find antiviral drugs that target the virus but don't target host structures as well. The most successful agents are these nucleoside analogues. AZT was the first of these back in late 80s, early 90s. This was introduced. This particularly targets reverse transcriptase in HIV, and this turned out to be a very effective agent. In fact, I think it's quite an accomplishment on behalf of the pharmaceutical industry and of humankind generally that we came up with these agents very quickly after the arrival of the HIV pandemic. There are also these nucleoside reverse transcriptase inhibitors. There are also these non-nucleoside reverse transcriptase inhibitors as well, which bind directly to the reverse transcriptase. They don't get incorporated into the growing chain of DNA, but they still nonetheless inhibit. There are now also a variety of other classes that have been developed over the years. Protease inhibitors, fusion inhibitors, both have used to target retroviruses like HIV. Influenza, we have amantadine and relatives, and these neuraminidase inhibitors. Again, these target a particular enzyme which is crucial to the life cycle of the flu virus. Another approach is to use things like interferons. These are the body's natural antiviral products that trigger antiviral defense pathways in the body, and if we apply these to uninfected cells, then they can provide some enhanced protection against viral infections. Fungi are also problematic because, like us, they are eukaryotes, and so finding selective targets there is quite tricky. You might not think it when you look at some yeast in the kitchen or whatever, but you're actually quite closely related to yeast. And much of the cellular machinery, the cellular toolkit that we use to run our cells is shared with those yeast. And so there are a number of targets, particularly the unique plasma membrane which these fungi have. There are these ergosterol inhibitors. There's the echinocandins that also inhibit the cell wall biosynthesis in things like Candida. And there are a variety of others there which are listed. As with bacteria and viruses, I didn't say it, but with viruses it's true as well, we do see the emergence of resistance to these agents. It's a little bit unpredictable. Sometimes agents work very well for long periods of time. Other times resistance arises fairly quickly. Okay, the last couple of minutes of this talk, I just want to move away from looking at molecules and cells to sort of look at a clinical perspective. So if a clinical practitioner wants to start using antimicrobial agents, what kind of questions do you have to bear in mind? Well, the first question that we ask medical students to think about, house officers, is does this patient actually need antimicrobials? As I pointed out earlier, many times a patient with a cold or flu will say, I want treatment, I want antimicrobials, I want antibiotics. They don't need them. Even when you're taking people, say, with chronic bronchitis, chronic airways disease, they may be coughing like a trooper, they may be wanting antibiotics, but things like physiotherapy are actually probably far more effective in actually clearing out their airways than giving them antibiotics. Half of the people that turn up in the GP surgery with what looks like a urinary tract infection, in fact, don't have any bacteria in their urine, and they don't need treatment as well. If we are going to treat, then we have to think, what's the best treatment? We have to choose an agent based on the likely organisms that we're anticipating to find there, and often you have to start treatment, so-called empirical treatment, before you actually know the identity of the organism. So you're basing it on what you know of that particular infection, what you know about your local infection rates and so forth. You need to look at how much, how often, what route for how long, how much does it cost. Now you might say, well, that shouldn't matter, but the difference in price between the most expensive antimicrobials and the cheapest ones are probably 3 orders of magnitude in terms of price. We have to ask also, are there any problems in using antimicrobials in a given patient? So the regimes that we would use in a child, for example, or in a baby, may be different from what we use in an adult. One of the least attractive features of my face is my teeth, and that's because I have what we call tetracycline banding there. So I was given tetracycline as a young child, and this has produced these brownish discoloration on my teeth. We don't now give tetracycline to children for that reason. Pregnant women also, we have to take special care there because the fetus may be susceptible to side effects from antibiotics that we wouldn't see elsewhere. At the extremes of age also, some agents that may have the ability to cause renal damage as a side effect, if you give it to an elderly patient whose renal function is a bit compromised already, you might have far more of a bad effect than you would if you gave it to a healthy young adult. The other thing we say is you've got to take samples before you give antibiotics, ideally. Otherwise, you won't achieve a specific microbiological diagnosis. That's a counsel of perfection. We know that the average general practitioner is not going to take a urine while the patient's there and then wait and then come back and wait till they report and then give antibiotics. They often just give antibiotics and see what happens. But as a counsel of perfection, it's much better to take the specimens first. So here, I've just already covered these points we're talking about the previous slide. So these are the sort of things that we need to think about. Do they actually need antibiotics? When we're looking at the kinds of antibiotics we're giving, we can sometimes make a very solid prediction. So if someone's got a streptococcal sore throat, we know that penicillin will work. The patient may be penicillin allergic, but it will work against the organism if the patient can take it. Most of the time, though, it's useful to take a microbiological sample and then do sensitivity testing because it will guide therapy. So if you have a urinary tract infection with E. coli, it's a toss of a coin as to whether that E. coli will be susceptible to the first-line agents like ampicillin or trimethoprim. And taking a urine will actually give you a clear view as to what agents you can use. And also, if you know that in your population that 75% of E. colis are going to be resistant to ampicillin and to trimethoprim, then you would say, well, actually, we won't even consider using those agents in any setting. If we look at the problems of antibiotic regimes in different patients, I've mentioned some of them already, pregnant women. We have things like allergy. So about 1 in 10 people in the population is allergic to penicillins. And sometimes they can be spectacularly allergic and actually develop what we call anaphylactic shock when they're given antibiotics. So that's a particular problem. Upset stomach is a common problem with many antibiotics. Erythromycin particularly chews a hole in your stomach. I know that from experience. Antibiotic-associated diarrhea. So many agents will cause diarrhea. And particularly in hospital practice, we often see overgrowth of an organism called Clostridium difficile in that setting. We get the problems of overgrowth of resistant organisms, and we get organ-specific side effects. So some antibiotics will specifically damage the kidneys. Gentamicin is an example of that. Some of them will damage the ears. Again, gentamicin does that. Some will damage the liver. So things like rifampicin, if it's given in large doses, might damage the liver. Chloramphenicol has this particular idiosyncratic side effect that in a very, very small percentage, less than 1 in 100,000 patients, it will completely destroy the bone marrow of the patient, and they'll develop what we call aplastic anemia. How much and how long do you give drugs for? So the key point, one of the key features we look for is the so-called therapeutic ratio. So that's how much of the drug do you need to give to achieve the effect you want, compared to how much of the drug would actually cause side effects, unacceptable side effects in the patient. So we need to find a dose which is effective but not toxic. Sometimes people are too far on the side of caution, and they don't like to give large doses because they think, oh, I'm worried about toxicity, particular problem with gentamicin, and so they underdose the patient. So in their attempt to avoid poisoning the patient, they actually end up not treating the patient at all. And when I used to work at the Royal Free Hospital, one of the consultants there used to summarise the problem with gentamicin, which was it was nephrotoxic, damaged the kidneys, and that people were scared about using it. He said there's nothing as nephrotoxic as death. So basically, if you don't treat the patient, they've got overwhelming sepsis, they're going to get their kidneys damaged alongside the rest of them when they actually become fatally ill. How long do we treat for? Usually we give them for too long. Patients get written up for like 10 days or 7 days, when perhaps 5 days of antibiotics is enough. Questions about how frequently, by what route. One of the things we try and do when we use antibiotics, if we're using them in the hospital, we try and get the patient off intravenous antibiotics onto the oral route as quickly as possible. So giving things intravenously, you know you're achieving predictably high blood levels, but it's a lot of faff and it's also expensive. And so we try and get off that as soon as possible. Last slide or two now, just so it's worth just flagging the idea that we sometimes use antibiotics prophylactically. I've mentioned this in the infection control lecture. So sometimes contacts of someone with infectious disease will get given treatment to prevent them from getting the disease. The other setting in which we might use antibiotics prophylactically is someone has a clearly defined increased risk of infection. So if any of you play rugby and you have a bad rugby tackle sometime and you end up rupturing your spleen and your spleen gets taken out, then that makes you susceptible to infection for the rest of your life, and in such a setting you will be given prophylactic antibiotics. I mentioned cost earlier. Cost is a problem. Usually new drugs are much more expensive than old drugs because when they're still within the patent duration of the patent, then the manufacturer can charge almost as much as they like, as much as the market will stand. Once they come off patent after about 20 years or so, then you can get anyone making that drug, and then there's a keen, lively marketplace of trying to push the price down. So maybe a company in South Africa or in India that's making it much cheaper than we make it in this country. I won't go through all of those points there. The cost broad spectrum more than narrow, IV more than oral. So we have to be careful in using drugs to avoid wasting money. Just the last point to make is that the struggle against bacteria is certainly not won. We may have pushed them into some extreme parts of our society. I don't suppose many of you go to bed worrying about whether you're going to get an infection or not. But there is still a problem. Multidrug resistance is on the increase. There is a risk that multidrug resistant organisms will return to menace our communities. And so we need new drugs. And so there are programs ongoing to try and develop new drugs. And there are lots of chemical methods, so-called combinatorial chemistry. You can screen millions of compounds very quickly, but you need to screen millions of compounds to actually get a single usable drug. So there's this whole pipeline that goes from discovering a a hit, a molecule that has some useful selective toxicity against a target, you then have to go through optimization, show that it can be absorbed, that animals don't die when you give it to them, then you put it into volunteers, and then eventually you show that it actually has the effect you want. That can take decades to go there. Okay, I think I've finished now. I'm not going to go through each of these things anymore.